Hi, I'm Ludovic Monin, and I will present now my poster, Phenotypic Variations of Primary Metabolites Yield During Alcoholic Fermentation in the Saccharomyces cerevisiae species, which is a part of my PhD thesis work on ethanol yield. The yeast Saccharomyces cerevisiae is the main actor of alcoholic fermentation, which is one of the main steps of wine fermentation. It can convert sugars from grapes, glucose and fructose, in ethanol and carbon dioxide with an outstanding yield of 92%. This ethanol production can be followed by other metabolites production like succinate, glycerol, acetate, or other minor metabolites. In my work, I study ethanol yield to understand how we can modulate it. We consider a screening strategy to assess this metabolic diversity, including 51 strains of Saccharomyces cerevisiae for diverse genetic backgrounds. We included 4 genetically modified or evolved strains for their low or very high ethanol or glycerol yields. After measuring each metabolite concentration and making a statistic analysis, we first took a look at the ethanol yield diversity. In the first figure, you can see the ethanol yield, expressed as gram of ethanol produced by gram of exhaust consumed. Each dot represents the yield of a strain with standard error as error bar. Each letter represents statistic differences obtained following a 2K test. We can see that there is huge statistic differences, but only between very extreme yield obtained from genetically modified strains or evolved strains. In the next figure, we can see the correlation between ethanol yield and glycerol yield on only natural strain. This correlation exists too when we consider all strains, including genetically modified ones, and is stronger. On the bottom left part of this poster, you can see the metabolic yield for minor metabolites like acetate, succinate, and alpha-ketoglutarate for each genetic groups. We can see that there is a clear correlation between genetic groups and these minor metabolite yields. For example, one yeast produces less acetate and more alpha-ketoglutarate. Rome yeast produces more succinate, and flor yeast seems to produce more acetate and less succinate. These characteristics are clearly linked to dedicated use of these strains in the natural niches. The bottom right corner of this graph, we represented the variation of each metabolite for natural on all strains, including evolved or genetically modified strains. We can clearly see that ethanol is a metabolite with the smaller variations that we can find in our analysis. This variation is even smaller when we consider only natural strains. Other minor metabolites, like succinate or acetate, show a very larger variation, amongst 75%. This work highlights 3 main results. The first one is that ethanol yields, even if it shows a very low variation, show a significant variation. Glycerol is confirmed as the best candidate to modify ethanol production, as it is always correlated with ethanol yield. Minor metabolites like acetate, succinate, or alpha-ketoglutarate seems not correlated with glycerol or ethanol, but are mainly correlated with genetic origins of the strains. Thank you for your attention, and don't hesitate to ask me questions at the seminar or to send me an email.